Welcome, everybody. On behalf of the genetics department, I'd like to welcome you all to the first Saunders lecture, our inaugural Saunders lecture here in the Babbage. So at least in terms of Cambridge history, the discipline of genetics is a very young one. But I'd like you to cast your mind back to those sepia days of the 1900s when people... Oh, shh. That was really good. I bit my tongue there. When people like Edith Rebecca Saunders or Becky Saunders were here in Cambridge. This was a time when, in this early 1900s, when the recently proposed chromosomal theory of inheritance was just beginning to be accepted by many, though not Bateson initially, I should say. And Cambridge was at the forefront of research exploring heredity and phenotypic variation. And indeed, in 1906, Bateson coined the term genetics as he was reconducting the work of Mendel that had been carried out around 40 years earlier. So when one focuses on the role that Cambridge played in the development of this exciting new discipline of genetics, it's names of men like Bateson and Punnett that are really at the forefront. They were tracking inheritance and variation in a range of characteristics predominantly in plants such as stalks and peas, and in Punnett's case in poultry. But if one actually looks at the publications that present their results, many of them are co-authored by Becky Saunders. So together, Becky with Punnett and Bateson, using large-scale, well-designed and carefully statistically analyzed experiments, they quantified many of the key genetic phenomena and coined many of the terms that we know and love today, like alleles, and in fact concepts like linkage. And I'd just like to comment on this linkage discovery, because when one reads that published work written in those sepia days, especially the series of reports on experimental studies in the physiology of heredity. What on earth is the physiology of heredity? These are reports to the Evolution Committee of the Royal Society. You can see that actually, rather like the publications of today, they outline at the beginning the contributions of the various authors and co-authors to the science that's in the report. The reports are lengthy. And in fact, it's very clear that while Bateson maybe looked at one or two plants and Punnett mated some chickens, Becky Saunders actually was working on many species of plants, and the data sets in these papers have predominantly been generated by her. So in one of the publications that I read recently, they report the discovery of the phenomenon of linkage, a phenomenon that they called coupling. She followed the segregation of hoary and glabrous alleles, in other words, the hairiness of plants, alongside the coloring of the flowers, and reported a preponderance of co-segregation between flower color and hairiness that suggested perhaps that these genes were not following Mendel's law of independent assortment. In fact, in her paper, she says, the preponderance of hoary individuals can scarcely be accidental. The results indicate certain linking together of surface character with flower color. The hoary character only appearing in conjunction with sap colors, the glabrous character only with white or cream. And she did controls. No similar association of seed color with surface character was apparent. So in modern parlance, the color and the plant hair genes are close together on the same chromosome and hence are co-segregating, and this is linkage. Saunders was also very much committed to the education of women scientists and was director of the Balfour Biological Laboratory for Women between 1890 and 1914, a building that actually still stands today at the back of the Downing site across from Wetherspoon's pub. It's a very characteristic building. And in that building, students from Newnham and Girton colleges received their preparation for the natural sciences tripos because they weren't allowed to attend classes with men. And at that time, indeed, that lab housed a team of quite remarkable women, including the biochemical geneticist working on plant pigment, Muriel Ownslow, who discovered epistasis using snapdragons, and also the comparative plant anatomist Agnes Arbour, who was later made a Fellow of the Royal Society. She was actually the third woman to receive that honour. She was made an FRS in 1946, and she worked on morphology and phylogeny. So more recently, Saunders and her women colleagues are getting greater credit for the contributions that they've made to genetics. And in this named lectures, we've chosen to celebrate Becky Saunders, not only because she epitomizes the contributions of unsung women scientists to the research in Cambridge in the early 20th century, when things were so challenging for women who were striving to make discovery, but also for her fundamental execution of really beautiful, quantifiable experiments that allowed her to elucidate not only the deviation from Mendel's law, but many other things too. And so it's a real pleasure to name this lecture the Saunders Lecture, and we hope to be having this regularly. Having said that, you'll notice that our seminar speaker is not a woman. And that is really because in selecting our speaker, we did not discriminate. We recognize that men, some men anyway, can do pretty good genetics as well. And actually, indeed in the department, we took a very democratic process to elect our first Saunders speaker. And our seminar committee collated names that were contributed by members of the department, and the whole department actually voted for who they wanted to have as their speaker today. Hence, we are very delighted to have Richard. So I'm not going to introduce Richard. Instead, I'm going to hand over to Chris Illingworth in our department. Chris is a rising star in our evolution and population theme. He's a Henry Dale Research Fellow who also chairs our seminar committee. And so, Chris, I hand over to you.
Thank you, Anne. Well, I'm delighted to be able to introduce our speaker, Professor Richard Lenski. Richard did his PhD at the University of North Carolina and then worked at the University of California in Irvine before moving to Michigan State University, where I've been learning today that he runs a very broad and diverse research group. Now, Richard is most famous for his work in experimental evolution using E. coli, and really has brought many things to light through that. In one sense, about mechanisms of evolution, thinking about the evolution of mutator phenotypes, and secondly, about the contingency of evolution. That is, the question of to what extent are evolutionary processes constrained or predictable, and to what extent are they sort of unpredictable and stochastic. In choosing a speaker, as well as looking for somebody with an outstanding research record, we also looked for someone who'd made a broader contribution to science. And Richard has a record of people coming through his lab to set up their own research, set up their own groups, and also has contributed to a broader understanding of evolutionary processes in a way that I myself appreciate, in that I use a lot of his work as part of my own lectures when I'm teaching this stuff to undergraduates and graduate students. Just in short, I'm delighted that Richard could be with us tonight, and I'd like to invite you to join me in welcoming Richard this evening. Thank you.
Thank you, Anne and Chris, for that warm welcome, and it's just a tremendous pleasure to be here. I've enjoyed meeting several of you and look forward tomorrow to more of the same, and at the reception, many more of you, I hope. So it's a real pleasure to be here. I'm really humbled by the Saunders connection, and I thought I would just put up a couple of things that echo what Anne said. So she was just critically important in the integration of Mendelian genetics into our modern understanding, And with Bateson and Punnett, as I read about some of her work, it was extremely interesting to me how much of it sort of foreshadowed what I think of as the integration of Mendelian and Darwinian thought. So even though what a lot of her work was critical at showing discrete variation, in their papers they talked about the fact, well, if you had a number of different, what we'd now say, genes contributing to that, you would end up with something that looked more like the quantitative variation that some of the other schools of thought were insisting the way thing always ought to work. So very integrative, synthetic ways of coupling what we now take very much for granted, but was tremendous discoveries in the day. Is my thing going to work? There we go. So that's just the point that I would, as an evolutionary biologist, would emphasize, was this laying the groundwork for the synthesis of these two very different schools of thought where people were battling which was the right way to look at it. In fact, her contributions, I think, very much underlie what became the sort of modern synthesis over the next few decades. Funny. All right. So, stepping over to Darwin for a moment, Darwin got, of course, many things right in his theoretical framework. So he was certainly not the only one. Lamarck and others had put forward the idea of descent, of a tree of life. But one of the things that he fundamentally got right was the fact that evolution had occurred and created these tree-like structures. Many of you have probably seen this, but I think this is just one of the greatest things from Darwin's notebooks, where back in 1837, not long after the voyage of the Beagle, he drew what we'd sort of think of as a simple phylogenetic tree, and above it he wrote, I think. He's formulating a hypothesis. You really see these very nascent ideas in his thinking. He actually, in his notebooks elsewhere, he coined the term unnatural selection, what we'd now call artificial selection, before he had come up with the words natural selection. It's really interesting. You see how science works in people's heads. And of course, he's most well known for, along with Alfred Russel Wallace, the idea of adaptation by natural selection. So again, these ideas were stimulated by the voyage of the Beagle, but this is some of the specimens that he and others collected from the voyage of the Beagle of what are now known as Darwin's finches, showing these different beak structures and beak morphology that were connected to the diets of these forms that had descended from an ancestral finch on the mainland. But he was wrong about some things. So his theory of inheritance would be more like what we might think of as an epigenetic theory. He had this idea of gemmules, and it actually is very Lamarckian in some ways, something circulating in the blood, and I don't know, it doesn't really fit with his own thinking, but it was the best he could do in the day. And of course, it took Mendel and Bateson and Saunders and others to sort this out. But another interesting thing was he thought evolution was too slow to observe. So even though the last words of The Origin of Species are about evolution and evolving forms, so that evolution is still ongoing, he actually wrote, and it isn't just in one place, there are multiple passages where he's sort of saying, but we can't really see the process. So he said in The Origin, he said, we see nothing of these slow changes in progress until the hand of time has marked the long lapse of ages. And then so imperfect is our view into long past geological ages that we only see that forms of life are now different from what they were before. So that has changed in recent years. We know we can see evolution both in natural populations, like Peter and Rosemary Grant's epic work on the Darwin finches in the Galapagos Islands, and in the right systems like fruit flies, nematodes, and many microbes, you can take this into an experimental realm to watch the process of evolution in action. And so the experiment I'm going to talk about today is called the long-term evolution experiment. And so I sometimes call it the LTEE. So if I refer to that, that's what it is. And it's this experiment it's a very simple experiment. If you come to my lab, you'll see just how simple it is. It's 12 populations of E. coli, all started from the same strain, except I embedded a genetic marker in it, so there were 6 populations that make white colonies and 6 that make red colonies. That color marker allows us to do things like compete bacteria from one generation that have one marker against the ancestor, which has the alternative marker, and because these are asexual, clonally reproducing organisms, there is perfect linkage between the marker and the rest of the genome. Anyhow, every day what we do is we take 1% from each of these 12 flasks and move it to fresh medium, which has as its sole limiting carbon source glucose. There's a second carbon source in the medium called citrate, which is present in there as a chelating agent. But E. coli, going back to its original definition, can't grow on citrate in the presence of oxygen. But it'll become relevant in the second half of my talk. But anyhow, this daily 1 to 100 dilution, if you think about the mathematics of that, that corresponds to about 6 2/3 doublings, 2, 4, 8, etc. And so that's how we calculate the number of generations over time. Not only are these asexual microbes, asexually reproducing organisms, but unlike bacteria, many bacteria in nature, there is no horizontal gene transfer. So there's no functional phage in the system and there are no plasmids. So mutations are providing the sole source of variation. If you do some back of the envelope calculations, you'd realize that every one of these flask lineages has had many billions of mutations. But of course, those are present only in one cell. Many of them will just go extinct by drift or because they're deleterious. And I'll come to the genomics later. And I switched from studying insects to bacteria because of the fast generations, among other things, but actually what I realize is this ability to freeze the organisms is one of the most amazing parts of this system, because what we have is a frozen fossil record of early on every 100 generations. More recently, we store every 500 generations. So when we have accidents, we don't have to start the experiment over. I wouldn't be standing up here today if that had been the case. So we can easily restart the experiment when there are accidents, and also we can compare things that lived at different points in time. So we can do direct competitions between ancestors and their descendants. So it turns out that we're now at 68,000 generations. A postdoc in my lab, Zach Blount, has put up the sign that every 1,000, somebody said we should do it like automated, but every 1,000 generations, he updates that. And it also turns out this Saturday we're going to have a party, because this coming Saturday will be the 30th birthday of the experiment. And so that little arrow is pointing to my entry in the first lab book of this experiment, time 0. And so a little anniversary coming up. So what was I doing with this experiment? And Chris in his introduction hinted at least one of those. So I was interested in kind of 3 sets of issues. One about the dynamics of adaptation by natural selection. Was it this very gradual Darwinian process, or was it more, were there temporal fluctuations in the dynamic, even in a constant environment? And how long could fitness increase before it reached some sort of limit? I was extremely interested in the tension between the randomness of mutation and drift on the one hand, and the sort of pushing deterministic force of natural selection in a constant environment. And so would these populations find their way to very similar solutions or to radically different solutions to this very simple environment we challenged them with? And I hoped that I would be able to in some way integrate the dynamics of phenotypic and genetic evolution, although as some grant reviewers early on pointed out, they had no idea how I was going to do it, and I wasn't sure either. But over the years, genomics, among other things, has come to our rescue in that regard. So what do the data look like in an experiment like this? So this top graph, what I'm showing is the fitness relative to the ancestor over just the first 2,000 generations and just from one population. So these are 95% confidence intervals based on replicate assays from that one population. The relative fitness is literally just the ratio of the growth rate of the evolved bacteria relative to that of the ancestor when they're competing head-to-head in the environment where the evolution takes place. Everybody's been in the freezer, everybody comes out of the freezer and is acclimated, sort of what a plant biologist would call a common garden kind of setup, and then they are mixed and we measure their growth rates while they're competing. And what you can see is that by the end of these 2,000 generations, this population grows about 30% faster than the ancestor. And I'm not going to go into the statistical details, but it fits a kind of a step-like dynamic that at first glance might be very surprising, but in fact, in an organism that relies on de novo variation to generate mutations, for most of the time when a beneficial mutation is spreading towards fixation, for those lucky few that make it to fixation, it's a small minority having no effect on the average property of the population. And then it's over a very short window that it suddenly goes from minority to majority, and that's what generates these step-like dynamics. So theory and simulation very much supported that, in fact, this should be our expectation, not an exception, at least in these kinds of systems. Let's step back now and look not at just 2,000 generations in one population, but look the grand mean fitness across all of the populations through 50,000 generations, and that's what's shown by those data. And the confidence intervals here are based on the statistical variability across the replicate populations, and I'll come back to that more later. But what you can see is by 50,000 generations, the average population is maybe 70% faster growing. But you can also see, well, it's really, really slowing down. And those two curves are showing two models. They're both about the simplest possible models you could have. One is called a power law, the other is called a rectangular hyperbola, and I'm kind of embarrassed to say that for a couple of our early papers, and even well into the Middle Ages of this experiment, I was fitting, assuming they were going to reach an asymptote that this rectangular hyperbola would give. There's got to be some limit to the growth. It made intuitive sense to me, like from quantitative genetics and so on. Well, they can't go on improving forever. So can we distinguish these two models? Because the blue model, and they both give very good fits overall to the data, but the blue model has no asymptote. It says fitness can go on increasing forever and ever, but at a progressively slower rate. And that's what this power law model has. So this is showing just one way of, you can't compare the two models. We did Bayesian fits, and the non-asymptotic power law fits much better. Many of you are more familiar with sort of squared deviations. So this is the cumulative squared deviation. So the blue model, the non-asymptotic power law, fits the data much better. But that's still kind of a data fitting exercise. Do either of the models make testable predictions outside the data set itself or within the data set itself? And this is, I think this is my favorite way of looking at these data. If you give your statistics analysis only the first 5,000 generations of the data, and you use this asymptotic model, from the 5,000 generation data, it will fit that lowest pink curve. Give it 10,000 generations, the bacteria have gone past that, what you thought was the asymptote, and they give you the 10,000 generations as the next curve up. Give it 15,000 generations, so it's constantly underestimating the future potential of the bacteria. The bacteria can go past what this very simple two-parameter model would predict. What's amazing about this power law model is you give it the first 5,000 generations of data, it fits the data perfectly out to 50,000 generations. More or less perfectly, I mean, as good as the data are. You give it the 10,000 generations. So it's actually not just fitting the data better, it has the power to predict into the future. And we've actually done some out-of-sample tests out to 60,000 generations, and it continues to perform much better. Now, you might say, well, this is crazy. Bacteria can replicate at ever faster rates. And what's interesting is you can project this out. So my goal is, okay, I'll have run this experiment to a first approximation 50,000 generations. I would like another person to take it over for another 50,000 and another person for another 50,000. So does this predict some crazy growth rate for the bacteria? Because we can, by the way, we can convert those fitnesses into kind of an implied exponential growth rate. So if you go out to the age of 1 million years, 50,000 squared generations, the predicted doubling time is 23 minutes. Now, some of you will say, well, wait a second, isn't E. coli already growing? And in a rich medium like luria broth or lysogeny broth, it grows at that rate. But in minimal medium, E. coli typically has a doubling time much slower. So it's not making crazy predictions. And in fact, if you extrapolate it to the age of the life on Earth, you're still in the realm where some bacteria in some media can grow about as fast as these projections are. And so it's just simply saying there are smaller and smaller increments to be had far into the future. What about the repeatability of the fitness dynamics? And as you'll see later on, I'm sort of glossing over an exception that'll come up later. But this is now just showing the data for one of those 12 populations, completely independent, by the way, of the other data I showed a moment ago. So this is just one of the 12 populations out through 50,000 generations. And all these competitions are taking place against the ancestor. What if we instead compete population number 1, which makes white colonies, against population number 2, which makes red? So how similar or different are they when they're competing against their contemporaries as opposed to against their ancestor? And that's what the data look like there. So now it's sort of arbitrary which one is in the numerator or denominator, but for a while the one in the denominator is winning in the sense that the fitness is going below a value of 1, and then it comes back up. And I don't want to get too much into these fluctuations, but those of you in the first row might say, well, wait a second, the fitness scale is radically different here. So if we put those same data on the scale that I showed relative to the ancestor, they are remarkably similar. the populations haven't diverged, again, with one exception I'll come to later, they haven't diverged by more than a few percent in their fitness relative to one another, even as they've gained like 70 or 80% relative to their ancestor. So at this most integrative, but also most crude level, evolution looks very repeatable. So when I started this experiment in 1988, there wasn't a single cellular genome that had been sequenced. It wasn't until 1995 that Haemophilus influenzae was sequenced, much smaller genome at the cost of I don't know how many millions of dollars. But over the course of this experiment, sequencing started to look like something, for a while we would sequence a few genes, a few candidate genes. But this is a paper that came out, not our first genomics paper, but one that I will sort of emphasize because of the data quality in it with Olivier Tenéon and others, Jeff Barrick and others, where we took each of the 12 populations, 11 different time points, and 2 individuals, or 2 what we would call clones, and sequenced their whole genomes, so 264 whole genomes. And this is the phylogenetic tree you get. And at first it looks kind of totally crazy. There's some that look like they haven't evolved at all, and others that have these really long things. And by the way, the ancestor is a point down at the far lower left, and time is running up this way. This is showing the same data, rescaling it, because we already knew at that point that 6 of the 12 populations had evolved hypermutable phenotypes. And one of the things that this genomic data made me feel very good about was I was always worried that we had had some cross-contamination between our populations. One reason for having these white and red colonies was that the instructions for whoever did the transfers was always do left, right, then on. But I imagine somebody had a really good party, like maybe we'll have later tonight, and they came in the next morning and they went down and did all reds, and that there'd been some undetected cross-contamination. But in fact, throughout this experiment, these 12 populations have their completely independent phylogenies. What about the rate at which mutations accumulate? So I'm going to now restrict the data only to looking at the non-hypermutable populations. So I'm not going to talk about the ones that evolved hypermutability. And what you can see here, this is, out of those billion-plus mutations that have happened in every population, how many are in a typical genome as time goes on? And so after 50,000 generations, a typical non-hypermutable population has on the order of about 80 mutations. And I've just fit two different models so far to this. One is, well, what would it look like if it was all neutral drift? And the idea there is that mutations would accumulate at a constant rate over time. And what you can see is there are too many mutations early on. We also have a dynamical model that is only natural selection, that gives rise to that power law dynamic that I showed before. So the theory also gives rise to that. And that generates this solid gray curve. And what you can see, if it was only beneficial mutations that were going to fixation, we would expect a curve we'd expect more mutations earlier and fewer later. But if you just simply combine these two processes, so nothing radical here, just drift plus selection, you can get an extremely good fit to the data. So the triangles, by the way, are the means of those populations. So it really looks as though there are driver mutations that are, by the end, roughly half of the total, and about half of them are hitchhikers or passengers. Those of you who've thought about molecular evolution might ask about, well, what about the signature, a classic signature in molecular evolution of the ratio of non-synonymous to synonymous changes within protein coding regions, and we adjust those calculations for the number of sites that are at risk for those two types of mutations. People often get very excited when they find an individual gene that has a ratio greater than 1 because that suggests a signature of positive selection, that gene has, in its history, has undergone selection to be different from its ancestral state. And what we see is across the entire genome, there's a very strong signature of that. Starting out in the first few hundred generations, basically everything is non-synonymous, but there are synonymous mutations, these hitchhikers accumulating over time, as well as some non-synonymous changes, which are presumably also neutral or nearly neutral. But even after 50,000 generations, we have about a 3 or 4-fold excess of non-synonymous changes relative to synonymous in the genome. So very clear, strong signatures of natural selection being important. What is changing? So if you look at the exact level of the nucleotide, there are very few of the mutations we find that are identical at the base pair level. But at the level of genes, there is tremendous parallelism. So E. coli has roughly 4,000 genes and about 4.5 million base pairs. Just 57 genes that comprise only 2% of the coding length of the genome have half, or a little over half, of all the non-synonymous changes we find in these non-hypermutable populations. So that's a very strong indication that natural selection is finding the same targets over and over. You can do a randomization test for this. I put this up only because it's my all-time favorite p-value if you do a strict randomization test. So anyhow, don't need to do that to convince with those kinds of numbers, as somebody I was talking to earlier today. What are the genes that are actually changing? And things are actually pretty complicated and interesting in this respect. Many of these genes, you might imagine that they're tinkering with kind of peripheral things that hardly matter at all. In fact, it's many very core metabolic and regulatory functions which are evolving, including pyruvate kinase. All 12 populations have changed the structure of that protein. There are 3 or 4 genes involved in cell wall biosynthesis that have been changing repeatedly. There are several genes involving in DNA topology that have been changing. The stringent response, where E. coli goes between sort of growth and starvation, has been changing. And some of these are essential genes. So this kind of signature looks like what we're seeing is a lot of fine-tuning of the regulation and expression of important functions, which might seem puzzling until you think about the history. You know, E. coli, even though it's been in the lab for a while, it's got 100 million years or something out in the wild where it's dealing with bile salts and competitors and viruses and all kinds of sort of contingencies of the environment. Whereas here we've put it in a very simple and predictable environment, and I think some of these genes that have been changing are ones that have the potential to sort of have good pleiotropy in the sense that they're changing, they're dialing knobs like in the stringent response in the cell to adapt very specifically to this cycle of starvation and growth that they experience in this environment. Interestingly, out of those 57 genes, there are 10 cases where at least 2 populations have mutations in the same codon. And in those 9, I forget whether it's 8 or 9 of those 10 cases, the mutations are in very close to known binding sites for proteins, RNA, or ligands. So again, it's consistent with this idea. What we're seeing is some very specific fine-tuning to the experimental conditions of this experiment. But there also are a lot of gene deletions. They're a little harder to interpret. Many of them are flanked on one or both sides by IS elements, so sort of regions where there's a potential for homologous recombination and that sort of things. But what's also interesting is if you look at the genes that are deleted, a large fraction of them are things that actually came in horizontally, not during our experiment because we don't have horizontal gene transfer, but during the history of E. coli. So remnants. I said there are no viruses, but there are sort of dead prophages in the genome, and those are being deleted repeatedly. There are plasmid-derived toxin, antitoxin systems that are thought to allow sort of plasmid stability by addicting the host with this sort of toxin antitoxin system. Those have been knocked out by non-point mutations in our populations. Also, things that are involved in adaptation to hosts, like fimbriae and extracellular capsules, those have been the sorts of genes that have been deleted. And again, it makes sense that those sorts of genes. What we don't really know is how many of them are beneficial and how many, because of this hypermutability associated with insertion sequences, how much it's mutability versus costs. But it looks as though they're eliminating unused and, at least in some cases, at least 2 cases, we've established costly functions. And the net effect is that the average genome size is declining, although, I mean, whether you consider that a large decline in 50,000 generations or small, I think depends on whether you come from sort of a molecular biology or an evolutionary biology context. I would view it as large, but some of my molecular biology colleagues, when I first started getting into the genomics, were expecting us to see something more like deleting half their genome or something. And it's clearly a much slower process than that. Despite the simplicity, I deliberately made this environment as simple as possible. Some fascinating complications have evolved. So I've already mentioned hypermutability has changed. And interestingly, we also see subsequent compensatory adaptations. So I'm going to kind of go through a little bit quickly on this. But this is where we, from one population, sampled 3 clones at 2,000, 3 from 5,000, et cetera, out to 40,000 generations. And this was before we had done the massive genome project. And we sequenced all of these. And these colors correspond to sort of different branch lengths, so it doesn't extend down the street too far. But I noticed that at 40,000 generations, oh, and this population, by the way, is one we knew had evolved hypermutability around this point in time. And I noticed by 40,000 generations, we had significantly different branch lengths, even in just synonymous changes. So that suggested that there had been a further speed up in the mutation rate that led to these two 40,000 generation clones, or perhaps a compensatory change that reduced the mutation rate along that branch. And as so often in this experiment, the answer turned out to be more interesting than either of those hypotheses, because in fact, both of those lineages had picked up compensatory mutations that reduced the mutation rate not to the ancestral level, but they were about a 100-fold mutator to about a 50-fold mutator. And interestingly, the compensatory mutations were in the exact same gene. So here we're seeing parallelism even between lineages living within the same flask. So not just parallelism across populations, but parallelism within it. And it turns out the mutation that's involved in compensatory changes for the mutation rate is one that biochemists had already worked out many years ago, but I knew nothing of that. But it was neat to see that we sort of recapitulated the results. It's changing not only the mutation rate, but the spectrum of mutations, so it leaves a very, very specific signature. Another thing that's changed is, even though I had intended this to be sort of a one niche environment, because I wanted to be able to talk about fitness in a relative sense, but in a constant environment. So I work at a very low glucose concentration because I didn't want lots of metabolites that would be changing the environment radically. And yet what we've found is that in fact, despite working at a very low glucose concentration, there are enough secreted metabolites that the ecology is getting very interesting, sometimes leading to lineages that coexist for essentially into perpetuity, at least as far as the experiment's gone, and sometimes sort of ecologically stable but evolutionarily unstable coexistence. So this is a case of where we found early on, before we had done any genetics, 2 colony morphotypes within a population, and you mix them at different ratios, and over the course of just a few, 100 generations or 120 generations or something, they converge on a stable ecological equilibrium. But if you look at that same population, not the ratio of those 2 types over these 20,000 generations, there are these wild swings, because just because there's a stable ecological equilibrium doesn't mean they're no longer evolving. They're evolving still relative to the medium and potentially co-evolving relative to one another, and that's driving these wild fluctuations. And yet, even at 50,000 generations, it's not really clearly labeled here, but even at 50,000 generations, this polymorphism, which rose probably around 6,000 generations, is still present in this population. Working with Ben Good and Michael Desai at Harvard, they said, why sequence these clones? Let's just take, they essentially wrote me one day and said, send us your freezer. They basically were proposing to sequence all of the samples, and not just clones, but to deep sequence the populations. And so that's what these data are. So we just had that appear a few months ago in Nature. So there's about 1,400 complete population samples here. Now there's still issues about depth of coverage that I'm going to gloss over. But what these graphs show is selective, hitchhiking and sweeps and the effects of drift. I mean, this is basically the dynamics of all these alleles. And so this is a population that seems to show sort of reasonable behavior of what at least I would have imagined when I started this experiment, of some nice, clean, selective sweeps, a little bit of mess in here. This is one that shows reasonably similar behavior, but then this is one of them that became hypermutable, so the whole thing becomes a total mess. But these are a very interesting one where we evolved 2 lineages that coexist, and they developed sort of a hidden Markov model that allowed them to sort of look for any cases by the correlate, because they're sequencing the population, they don't always have the linkage relationships. And so what they're using this model to do is to essentially say which alleles are going up and down in concert with one another. And this sort of summarizes it over on the right, but basically it looks like 8 or 9 of the 12 populations have had either transient or sustained coexistences, but when I say transient, at least 10,000 generations. So there have been periods of ecological stability followed by periods of extinction of not just individual mutants, but of clades that have accumulated many mutations in these populations. So I'm going to give a partial summary before I sort of shift gears for the last 15 minutes or so. Most of the observed mutations, excluding the hypermutables, are beneficial. And despite the increasing complexity, we see substantial parallel, the complexity of this ecology and of these hypermutables, we still see substantial parallelism both phenotypically and genotypically. So it's almost as though in sort of a Sewell-Wrightian framework, to cross back the ocean, the populations are climbing Mount Glucose. They're not doing it identically. They all start from an ancestor that's not particularly well adapted. You know, some are taking the southwest face and some the northwest face, so to speak, but they seem to be, I was expecting the results to be much more divergent. But there was a striking exception, which is around 31,000 generations into the experiment, we come in one day and we find one of the flasks. This is, by the way, at stationary phase because we don't put so much glucose in. The normal populations have a density of about 5 times 10 to the 7th cells per mil, so much lower than most microbiologists would work at. And one of our populations was much more turbid, so I thought we had gotten a contaminant that was able to grow on citrate. There are many bacteria that can grow on citrate, and I said we better start this population over, and in the meantime we began checking this out, and as we checked it out, we realized, no, it's an E. coli, and it's an E. coli that is derived, the markers showed that it was derived from our ancestral strain, and not only was it derived from our ancestral strain, it had the markers appropriate for that population. And I had a very talented postdoc in my lab, Christina Borland, coming from a molecular genetics tradition, who was going to try to map this, and she was having a terribly hard time. That could be because it was a big genetic change that wouldn't like easily get packaged into like P1 for transduction, or maybe it was a multiple sites, that there had to be sort of some potentiating mutations as well as the actual import mutation. So we weren't making much progress. And genomics hadn't quite reached the point that we could sort of immediately tackle it with that, although I'll come to that in a minute. But with Zach Blount, a postdoc and then a graduate student and now a postdoc in my lab, we sort of said, can we ask this in sort of an evolutionary way? Is it that it's an extremely rare genetic event that doesn't scale with our understanding of point mutation rates? Maybe it's an inversion that had to be exactly these two endpoints. And it could have happened in any population at any point in time. And the alternative hypothesis was maybe it's a perfectly ordinary mutation, but you had to have the right genetic background in place before it happened. And the answer turns out to be a mixture of those two. But this is Zach Blount with about 1/3 of the Petri dishes he used in this experiment. The idea, we originally wanted to replay evolution in the flasks just as we had, starting with different time points. And again, we're starting from clones, not from the population, because our question is about the potential of a mutation to allow the invasion of this this available resource. And it quickly became clear it was going to be very difficult to do. So what Zach would do instead is he would take clones from different points in time in the history of this lineage, grow up large populations of like 1 billion cells, spin them down and plate them on a medium that only had citrate, and incubate them for a month to see whether he could find anything. It turns out, because all these things obviously can't be dealt with at once, the experiment had to run over multiple years and very large. I sometimes put up there, I think he has the tightest confidence interval around the value of 0 in the history of genetics, because from the ancestral state, he tested 10 trillion cells, and not a single one ever evolved this phenotype. But after about 20,000 generations, you had a genetic background where, although it was still a very rare event, he found 19 additional citrate-positive mutants. And the statistics show that there was a change in the genetic background that created this potential to evolve it. So what it's saying is, in fact, there is a historically contingent process in the sense that as the populations were climbing Mount Glucose, at least one of them, and potentially in the future we'll find others, but even after 60-some thousand generations, there's still only this one. One population found its way into sort of this and this kind of makes it a cartoon, like it's very simple, but out of all these genotypic variants, one found the way to where it was now accessible by 1 or perhaps 2 mutations to begin growing on the citrate. So we then started, the genomics era entered, the cheap genomics for NSF budgets entered, and we began sequencing from this population. Some of you may say, was this one of the mutator populations? And the answer was no, it was not hypermutable, although it became hypermutable after it evolved the ability to use the citrate, but it wasn't before. And we were able to identify this clade where all of the new functions had evolved. Other people have been working on what some of these background changes are, and that turns out to be a very interesting story. We call that potentiation. And interestingly, it looks like there was sort of a reversal of a couple of mutations where they first came on and then subsequently disappeared, which made it a little difficult to figure out. But Jeff Barrick at University of Texas has led that work. What I just want to show you in the last few minutes is this, what was this mutation that first made a cell able to grow on citrate in this population? And it was a duplication event. And at first you might say, well, a duplication might be a dosage effect, a little bit more of just enough to eke out some growth. But what we were able to show was, in fact, only the second copy matters. And the reason is that there was a promoter downstream of the first copy which has been co-opted and which allows extremely weak but enough to kind of gain a foothold growth. So it's only the second one that is involved here. And so it's sometimes in literature this is called rewiring or promoter capture. And that was the event that allowed this population to begin growing on citrate on the right genetic background. You might say, well, what happened in those replays? I said there were 19 replays. Did they all do it the same way? And interestingly, at one level, they all did it the same way, but none exactly the same way. So all of them were physical recombination events. All of them involved this CYT gene, which, by the way, is the anaerobic transporter for bringing citrate into the cell. And some of them have co-opted IS elements. Others are actually, I think one of them is a deletion that brings a distant promoter upstream, et cetera. So anything, not anything, but at least the right promoters in context can give rise to this function. But clearly no single point mutation was able to do this, or it would have happened over and over and over in all of the populations. So I could stop there, or I could give a little bit of work in progress for maybe 5 minutes. So Zach Blount is beginning to sort of ask this question about speciation. So is there any sense in which the citrate users are a new species? And our interest in this is not naming anything. We're not it's not about the name of a species or is this a species. It's about, as an evolutionary biologist, asking for general principles about what we mean by speciation. So this is a discrete novelty, unlike, say, just growing faster. On the other hand, lots of things are discrete novelties, like phage resistance and antibiotic resistance, so that's not very satisfying. But it's a meaningful one in the sense that it contradicts part of Escherichia's definition of E. coli, so that makes it interesting. And I didn't go into it, but it's also interesting because it's cladogenic in the sense that it isn't just that the citrate user replaced its progenitor, the citrate positive and citrate negative lineages coexist. But those of you who work on plants and animals will say, well, what about biological species? And this is from Ernst Mayr, and it really is reflected, I think, in some of Darwin's writings as well, is that it all hinges on sort of the ability of our groups reproductively isolated from one another. Are they interbreeding? And if they are, then they are the same species, and if they're not, then they could be different species. And with some caveats about this being in natural populations, because lots of things can hybridize in zoos and so on. So at first glance, you say, we can't possibly address this. Bacteria are asexual, and there's, as I said, no horizontal gene transfer in the long-term experiment. But what if we set up a set of side experiments where we introduce horizontal gene transfer? Would we see a barrier to gene flow? And the kind of barrier that we think might be relevant here is sort of an ecological inferiority that implies that maybe as the citrate users get better and better at using citrate, and by the way, they still use the glucose, but they're not as good at the glucose as the glucose specialists, maybe sort of the intermediates would be less fit. And that would imply that if they were able to recombine, if there was horizontal gene transfer, there would actually be sort of an ecological barrier to recombination. So the idea is that as it continues to evolve on citrate, it would become more and more distant in a fitness sense from the ancestor. So to test this, and Zach is trying to generalize, we're really only going to look along one genetic axis. You might say, why don't we take the citrate out of the medium and ask it there? The problem is citrate is there as an iron chelator, and so that would change the environment in ways we don't want. So what Zach has done, first of all here, is simply showing, instead of competing things relative to the ancestor, he's competing later citrate-positive clones against the earliest citrate-positive clones. And yes, they are continuing, not surprisingly, yeah, right? When new inventions in evolution or humanity are discovered, things, that wasn't, the first airplane wasn't a 747 or whatever, there's lots of room for improvement. But what he could do is take those same clones that were getting better at citrate and essentially pull the rug out from under them by replacing that tandem duplication with just a single copy of the gene. In fact, we didn't even need to use recombination for it because duplications are prone to recombination. We could find a fairly high rate of going back to a single copy, and those that had the original promoter couldn't grow, but they would have, across these later generations, all the mutations that further adapted them to growth on citrate. And the prediction would be they would be falling in their fitness if you pulled out their rug from under them, and sure enough, that's what's happening. So it looks as though this seemingly irrelevant but neat evolutionary concept of reproductive isolation, even in certain very specific conditions, could be highly relevant to bacteria. So I'll stop there and summarize. I think I've mentioned all these things, so I won't read through the summary, but I do have an experiment that's been going on for 30 years as of this coming Saturday. I'm obviously not doing the transfers. I've been really blessed with having 3 amazing technicians consecutively, not all at the same time, over the years, including currently Nirja Hajala, who's been with me for about 20 years. And this is only a partial list of collaborators, grad students, postdocs, undergrads who've participated in this project, and so all the thanks go to them. I've only mentioned a few of them, but they've all contributed very meaningfully. So I'll stop there, and if there's time, to try to answer a few questions.